Is I'm going to kind of, instead of having it fold back, right? So instead of having this folder and drawing it with both halves here, we're going to kind of open it up and look at each part a little more individually. So trying to simplify that a little bit. Down here, I'm going to draw a big blood vessel. Okay? And that blood vessel is going to go into, it's going to draw a little thing that looks like a ball of yarn. We'll talk about why it does. Okay? And then coming out of the top, I'm going to draw another blood vessel. I'm going to draw this blood vessel more narrow. So narrower. The top one is going to be not as wide diameter as the bottom. Okay? These two structures are not part of the nephron technically. Okay? But this is what brings the fluid into this tuft of capillaries. So this is called the afferent. Remember, afferent means to carry towards arterial. Okay? Now, arterioles, we said, always lead to where? So the next structure would be a capillary, right? So this tuft right here is a tuft of capillaries called the glomerular capillaries. So here's our glomerular capillaries, this little thing that looks like a ball of yarn. And in fact, if you translate glomerular capillaries, it literally means ball of yarn. Because if you look at it under a microscope, it looks like a ball of yarn. They're all these intertwined capillaries. We'll draw some more structures in a second on that. Now, we said the kidney was a portal system, right? So we have a second set of capillaries later. This is the only place in the human body where not only do we have two capillary beds back to back, but at the same time, we have two arterioles back to back, which is totally strange, right? So leading from this capillary, these glomerular capillaries, which are often just easier said to be glomerulus. So glomerulus is just a singular name to say glomerular capillaries. We have the efferent arteriole. So leading out of that is called the efferent arteriole. That means to carry away. But it's the only place where we have an arterial bed right after a capillary. So we have arterial, capillary, arterial, and then we're going to have another capillary. I'm not going to draw them in, but they're kind of shown up there. I'm going to draw an arrow. I'm going to put two what's called the peritubular capillaries. Okay, the peritubular capillaries are the capillaries that go and intertwine and serve the tubules. Okay, the tubules are these tubes, right? You have a whole bunch of these tubules that the urine eventually carried through. Peritubular capillaries are the normal capillaries. They do the exchange. So they exchange especially fluid, ions, stuff like that. So the peritubular capillaries, their job is exchange. And so peri means around or close to, nearby. And so the peritubular capillaries are nearby the tubules, so we can exchange stuff. Again, if it stays in the tubule, it's going to eventually come out as urine. So if we need to pull water back, we need to pull sodium back, we need to pull glucose back, we need to put it back somewhere in the blood somehow, we do it through the peritubular capillaries. That's its job. Okay? The glomerulus then has a different job. It's not for exchange, it's for filtration. So the job of the glomerulus is filtration. It is built for its filtration. We'll talk about all the different attributes it has structurally that supports its functional process. Its functional process is what? Filtration, not exchange, filtration. Now, just to make life a little easier, I'm going to draw a little circle around my glomerulus. And that circle is going to kind of come together. I'm going to draw some squiggly lines. Then I'm going to draw something that goes down and comes back up, looks like half a paper clip. I'm going to draw some more squiggly lines. And then at the end, it's going to open up to a big structure. Okay? So I've drawn a circle around the glomerulus. That's going to be a structure we're going to talk about. Some squiggly lines, a half a paper clip, some more squiggly lines, and then a big tube. Okay? So we'll kind of identify these structures I just put in, and we'll look at them a little more closely. Okay? Now, like many things in science, you know, they're trying to go away from these proper names. You know, again, I think we mentioned it before, like your station tube, right? I don't know what that is. Well, if I say auditory tube, it has something to do with the ear, right? So at least you kind of know where it is. The problem is that people still use a lot of different names. Like, you know, to be honest, I don't even know what the scientific name of, there is one, for the Golgi apparatus, right? But Golgi's named after Golgi, right? But it's still a proper name, and everybody still calls it the Golgi. It's got a scientific name. I don't even know it is used so infrequently. Well, the same thing for this. This structure right here on the outside, okay, is called Bowman's capsule. Occasionally, it's called, but not very often, it's called the glomerular capsule nowadays. But Bowman's capsule is by far the most common thing. And Bowman's capsule actually has two layers. It's got this outer layer that you can see right here, this line. And it's got an inner layer that actually sits right on top of the glomerulus. Okay? So we'll talk about those in a second. And they're named the same way your pleural membranes are named. So like remember your pleural membranes or your peritoneal membranes, remember the serous membranes that you have? So there were two sets of them, right, in each place. The first set was lining the cavity, and the second set was literally on the organ itself. Anybody remember what those names were? Visceral and parietal, right? So it makes sense, the visceral one is on the organ, okay? So the parietal one's the outside. So this outer layer right here, that's the parietal layer. Okay, so that's the parietal layer right there. And in addition, you have it right on top of the glomerulus. The one on top of the glomerulus is the visceral layer. And we'll talk about what that does and what that is in a few minutes. Okay, so to kind of make a quick summary, all right, and I'll erase this top of the board and put it back up. The nephron is divided up into two structures basically, and the first structure, so the nephron, the first structure is made up of what we call the renal corpuscle. And the renal corpuscle has two subparts, the glomerulus and Bowman's capsule. And Bowman's capsule has two subparts, the visceral and the parietal. Does that make sense? So the renal corpuscle is part of the nephron. It's one of the two principal parts of the nephron. It's got two parts. The glomerular capillaries, its job is to filter, right? Known as the glomerulus. And Bowman's capsule, which is called the parietal layer and the visceral layer. So it's got two subparts to it. So let's take a look, a little more detail at the glomerulus and in fact then Bowman's capsule, what happens. Okay? So for the glomerulus, I'm going to draw it up here. The glomerulus is a capillary. So first of all, we're just going to draw kind of like we did before, this little capillary, this little vessel. Okay? One of the things that you want to do if you want to be a good filter is you need to be permeable, right? And one of the ways that we make the glomerulus permeable is it's a fenestrated capillary. Anybody remember what that means? Pores. It's got holes in it, right? So it's got a whole bunch of holes in it. So it's got fenestrations. Matter of fact, when I remodeled my house, right, I didn't buy windows, I bought fenestration, which literally is a German word for window. That way, because fenestrations, sounds more fancy, you can charge more for a fenestration than a window, so I think I paid more for them. But they're fenestrations, so they're holes, okay? What does that do? It makes it more permeable, right? So it makes it more permeable. So fenestrations makes it more permeable. So it's much more leaky than a normal capillary. The problem is With these big holes, it's like, getting this big, huge, colander with 4 holes in the bottom that are huge and dump spaghetti. And what's going to happen to all the spaghetti? It's just going to fall through, right? It's not a good filter yet. So we have to add to that. We have to prevent stuff from getting through. And so, this is a normal capillary. Capillaries are made up of what, typically? Simple squamous epithelium, right? Simple, one layer squamous epithelium, that's it. Well, all epithelial tissue has what's called a basement membrane. The basement membrane is what holds everything together. It's like taking a piece of Scotch tape and putting paper on top of it. The Scotch tape would be the basement membrane. That would be holding the paper, right? The paper would be the simple squamous cells. Well, the same thing happens here. This has a membrane on top of it, okay? And it just sits right on top, and it's called the filtration membrane. You have different names as well, and sometimes people collectively use the term filtration membrane, but that's not a bad name. It's the basement membrane of the tissue. The basement membrane actually acts as a filter. It's charged, and it keeps proteins and other charged molecules. It makes it a little more difficult for them to get through. So it's kind of like taking that colander that has the big holes and putting a cheesecloth on top. Cheesecloth is like gauze, basically, right? So it helps to protect it. So it makes it still really permeable, yet now we start to get a better filter. The problem is that's not good enough. And again, here's the problem we have, okay? How many red blood cells do we have? 5 million per microliter of blood, right? And we just said earlier, we filter 180 liters of fluid a day. So basically, your cardiac output almost 40 times. If you filtered and you got red blood cells into your urine, you would either A, pee them out, right? Or B, have to take time and energy to bring them back in. And it's just like your parents told you when you were a kid. Hey, it's really easy to clean your room if you never let it get dirty in the first place, right? Well, it's really easy to not worry about red blood cells and white blood cells and platelets if we never let them filter into the kidney in the first place. So we have another apparatus that helps to keep larger molecules from getting through, okay? And that's the visceral layer of Bowman's capsule, okay? What happens with the visceral layer of Bowman's capsule is it sits right on top, okay? Right on top of the glomerular capillary, and it has a special cell, looks like this. Looks kind of like the old Pac-Man, if you ever played Pac-Man, right? The little ghosts when they turn into ghosts. And they have these little feet, and then what happens is you have another one that sits right with it and intertwines its feet with another one. So basically, if this hand is the one on top, right, it's got the little feet, and this hand's the one on the bottom, they do this. And they intertwine, and they get these little tiny slits. And the only thing that can go through those little tiny slits are small enough molecules like water and certain ions that can fit through. Larger molecules cannot physically get through because the space is too small, okay? So, not surprisingly, these things that have little feet are called podocytes. What does podo mean? Foot, right? You got a bad foot, you're going to go see a podiatrist, right? They look at feet. So podo, foot cell. And the little feet of it are literally called pedicels, which literally means little foot. So pedicels are these little tiny feet that intertwine. And what happens is what's left between them, the little space, is called a filtration slit. So the space the fluid comes out in between the little feet, right, the little holes, those are called filtration slits. And those are really, really small. And what happens is it's just physically too small for things to not get through that are big. And the 4 big things that we worry about are all the formed elements, which would include red blood cells, white blood cells, platelets, and proteins, which are too big. So those slits are too small to physically allow the other molecules to move, those molecules to move through, okay? So that's going to become important, obviously, more in a minute, but it's important. So our podocytes are really just the specific name of visceral layer of Bowman's capsule, right? So the podocytes make up the visceral layer of Bowman's capsule. So if my arm was the glomerulus and my hand was the podocyte, they sit right on top and protect, again, from filtering too much stuff. Ultimately, now there's also, because these are modified epithelial cells, there's also a membrane that sits underneath it too, and that's called the slit membrane. Bottom line is we have a capillary that's very permeable because of the fenestrations, right? So we have it very permeable, but we also have it what we call selective. It's because of the filtration slits and the two membranes that make it more selective. Does that make sense? So we're still really, really permeable. Depending on who you read, a glomerulus is 100 to 1,000 times more permeable than a normal capillary because of the fenestrations, but much more selective because of the filtration slits and the slit membrane and the filtration membrane. What?
I can't see it. What does it say underneath filtration membrane? I just can't read it.
Slit membrane.
Oh, thank you.
Slit.
I'm trying to just...
Yeah, so there's two membranes that are important. Each one are associated with the epithelial cells, the filtration membrane and the slit membrane, and then the little filtration slits. Together, those three things make it more selective.
Why not have a continuous capillary? That way you don't need any of that.
But you need stuff to get out, but you have to be very, very permeable because we need to filter all that fluid a day, right? And the problem is if we don't have that selectivity, then we'll be losing stuff that we have to either lose or to figure out a way to take back in. So this seems obviously, evolutionary-wise, to be the best.
So the podocytes that continuously wrap around.
Yeah, so continuously wrap around. So there's a whole bunch all everywhere. As you look at it, they're literally look like they're painted over it. So all the way along the capillary. Remember, these are all intertwined and go all over the place like a ball of yarn because that increases the surface area, gives us more times for filtration, right? If it was just straight, we'd have a very short time for filtration, but because it wraps up and goes, back and forth, we have these, huge amount of space, volume in that space, and that allows it to more filter. And the bottom line is, because of the way it's set up, we're very, very selective, yet we're also very, very permeable, and it works out great in terms of where our kidney works. So, not surprisingly, and this is kind of get to the idea of the selectivity, the stuff that gets through that's been filtered is called the filtrate. Don't write that down yet, because it's not called the filtrate yet. Okay? Because it's protein and cell-free, we use a different term. We call it the ultrafiltrate to say, hey, it's free of proteins and the formed elements. So ultrafiltrate is what gets through. That's the name we call it physiologically. Okay? So the liquid that gets filtered through, which is normally has 0 protein, 0 red cells, 0 platelets, 0 white blood cells, is called ultrafiltrate. Obviously, when it's in the capillary, it's part of the blood. What's the liquid portion called then? Plasma, right? The major difference between plasma and ultrafiltrate is plasma has proteins, ultrafiltrate does not, right? It stays ultrafiltrate as we move through the tubules, and we'll see until we get here. We'll talk about this part. It's not part of the nephron either. Okay, so this is the exit. We'll talk about that. So, understand the different parts of the glomerulus, right? And the part that makes it more permeable, besides the capillary, it's already designed to be leaky. It's got the fenestrations, the windows, which make it even more leaky, and the parts that make it more selective, the filtration slits because of the podocytes, which is actually just visceral layer of Bowman's capsule, and the membranes that make up those two structures. Yeah.
So if there is blood in your urine, then it's basically because of podocytes?
No, probably not. I would guess that for most of us, 99% of the blood in the urine comes after.
Oh.
Most of the time from the bladder. People who exercise, very common for them. It's called hematuria. So hema, blood, churia, urine, basically. Hemato, right? People who exercise, runners, cyclists, right? Just from the vibration. Most of the time, an example, when they go out to exercise, the last thing they do before they leave their house is they go pee. So your bladder sits normally, in this cavity, and now it's just a little deflated balloon. And so it just bounces up and down, gets a little irritated. And when it gets a little irritated, you get a little bit of bleeding. It's microscopic, you probably can't even see it in terms of that. Obviously for women, menstrual period, or if someone's got a UTI, urinary tract infection, right? That's going to cause irritation and bleed. As a matter of fact, some people, that's how they know they got it, right? It's, urine turns orange or something. So, that's an issue. You could also have other things that cause it, even, bad shocks on your car driving down to LA. It doesn't have to be that far. But so there's a lot of different things that can go into it. If this is the problem where this is not a good filter and you're getting, blood in your urine, that is a major issue. And that's going to have, major clinical and physiological complications. And you're heading towards, severe renal disease, and if it, gets too bad, you're going to need renal transplant, dialysis, things like that, because you're no longer really a selective filter. That's typically not the case, but that's why you always say, and we'll talk about it in lab a little more, all that stuff we measured in lab, we normally shouldn't see in urine, but guess what? There's all normal reasons why we might see it. But if it is not that normal reason, then it could be something that's major, and that's the problem. You can't say, oh, it's probably just a UTI, or it's just because I ran 8 miles yesterday, right? It's just, if there's blood coming from someplace it's not supposed to, you should probably get it checked out. But most of the time it's not a big deal.
Kind of related, I've also heard like runners or athletes, if you take Motrin and then they become like a little dehydrated, they can like damage their kidneys, blow up their kidneys, because I guess the Motrin goes through there and it gets clogged or something, or?
Yeah, as you dehydrate, right, it concentrates that, it makes it more difficult. Our kidneys aren't very good at dealing with a lot of drugs, to be honest. Our kidneys are a lot tougher than people think. When people start eating the high protein diets, right, and we said one of the functions of the kidney is to get rid of the nitrogen waste, and a lot of nitrogen waste comes from high consumption of beef, especially. So when people start going these, high protein diets, Atkins type diets, a lot of doctors said, hey, these people are going to have kidney failure. Everybody's going to start having kidney failure because our kidneys can't handle it. Well, some people did, and maybe because of their high protein diets, but they already had, kidneys that were already bad. So it may put that kidney already bad over the edge. But a normal person with normal kidney function, they can handle that load, no problem. And it's probably true of most people with the same idea with the Motrin or Advil, any ibuprofen. Other drugs do it too, but there is a slightly higher risk because of that. But again, it's typically people that already have kidney problems. It just accelerates it in terms of that. But it certainly could be an issue. So that is our renal corpuscle. Okay. The other part of the nephron are our renal tubules. So we'll kind of go up here. So the second part are our renal tubules. And we'll list them first and then kind of put them on the thing. The first one is called the proximal, okay? Convoluted, that's what we kind of tried to draw a little bit of zigzagged line, tubule. Okay, so the proximal convoluted tubule. Again, because we're lazy in science, we just call it the PCT. Proximal convoluted tubule. Okay? Then we have what's called the loop of Henle. Again, another much more common usage. Sometimes it's called the nephrotic loop. Okay? So the loop of Henle, and it's got a couple different parts to it. It's often just called LH, but I don't like that because then it's going to be confused with like luteinizing hormone. Do a lot of stuff that has the same initials, always a problem. And then, not surprisingly, if you have a proximal convoluted tubule, you have a distal convoluted tubule. And not surprisingly, the proximal convoluted tubule is PCT, the distal is DCT. If you use abbreviations on an exam, make sure you are clear. So if I said, hey, what's the tubule closest to the glomerulus? We haven't even identified it yet, but you could probably figure it out. Which one do you think? PCT, right? If you wrote this on exam, I would mark it wrong. Right? So hey, it's a D. No, it's a C. It's a P. No. Right? Write it out or be clear. If I can't understand it, I'm assuming you're wrong. So, you know, think about that. So obviously, this first zigzag stuff right here is called the PCT, proximal convoluted tubule. All right. Obviously, here again, they've shortened this considerably, right? But it's much, you know, more zigzaggy than even this. But that's the initial one, and it goes down to Lupen-Henle and then comes back. So the one that looks like the hairpin turn, I'm just going to put LH in the middle. That's the Lupen-Henle. This structure right here is the DCT, distal convoluted tubule. And then this part right here is not on this list, okay? So this part is called the CD, which stands for collecting duct. The collecting duct, as you can kind of see up there, drains a whole bunch of nephrons. So that's kind of like the common drainage pathway. The collecting duct is not part of the nephron. Okay, it's not considered part of the nephron. Largely because there's not much physiology that happens into that. Okay? Since we're no longer in the nephron, we changed the name. Right? So let's go back to the beginning. The liquid inside the glomerular capillary, inside the capillary, because it's in the capillary, is plasma. And then the liquid here that gets filtered through because it's protein and cell-free is called ultrafiltrate. And it stays ultrafiltrate all the way through the nephron until you get to the collecting duct. Once you get to the collecting duct, we actually change the name to urine. So it becomes urine because we're no longer in the nephron, and most of it is pretty much, nothing happens in the collecting duct for the most part. So it's leaving as urine. This is stupid, but this is how I remember it. Once you get to the collecting duct, you're in. So urine's in the collecting duct. All right. So let's talk a little about the PCT first, the proximal convoluted tubule. Okay. It's got a bunch of cells that do certain things, but and this kind of analogy, it's stupid, but it makes sense to me. You go shopping, right? And when I was a kid, you know, I used to go out and help my mom taking groceries. Why? Not because I was nice, because I get the good stuff first, right? You know, in my family with 5 brothers and sisters, 6 of us, you know, if you wanted a Twinkie, you needed to get it the first 5 minutes you came home, right? So the good stuff, right, you want to get early. And that's what the proximal convoluted tubule is primarily designed for. The proximal convoluted tubule does not all of it, but most of the reabsorption. This is stuff that we're going to take out of the tubules and back into the body. Does that make sense? So the proximal convoluted tubule is primarily concerned with reabsorbing stuff. Taking stuff back in, we just don't want to pee out. Okay? And we do it first so that we make sure, hey, I got it, I'm good. So it happens primarily for reabsorption. Some of the reabsorption is passive and some of it is active. So not surprisingly, if you have active transport mechanisms, you need lots of energy, so you also need lots of mitochondria. So the proximal convoluted tubule is lots of mitochondria, and its job is primarily for reabsorption. We'll talk a little more about it later, but that's good now. Now the Lupen-Henle has 3 different parts to it. And each part is very different in terms of what happens physiologically. The part that goes down, okay, not surprisingly, is called the descending limb. So the descending limb is the part where the fluid goes down. The descending limb is very unique in terms of its structure. And what the descending limb does basically, or probably the easiest way to say it, it's permeable only to water. So the only thing that can move across the descending limb is water. This is a great system because it allows us to move water without worrying about solutes. Okay? There's something we haven't talked about it yet called bulk flow, or what's called solvent drag. Okay? They're the same thing, but they're opposite, but just we don't have to know this per se, just kind of give you the idea. If I have plasma and I move the plasma, because things are dissolved in it, what happens? They move along with it, right? So that's called bulk flow. Solvent drag is if I move a sodium molecule, right? What moves along with it because of its osmolarity? Water. It's hard to separate the two, but it's nice because I may want to get rid of a lot of water, but not any other thing that's dissolved in it. So by having the descending limb permeable only to water, I can adjust how much water there is without having to worry about obligatory movement of other things. All right? So it allows me to fine-tune stuff. Not surprisingly then, the descending limb is very important for concentrating or diluting urine. Okay? Now what's interesting is we have different descending limbs, or different nephrons in general. Some are really short like this, and some are medium, and some are really long. Which one do you think of A, B, and C would be best at concentrating and diluting urine? C, okay? So let's do a little quiz. Let's look at a fish, a kangaroo rat that lives in the desert, and a human. Which one's A? Fish. Why? Do fish need to concentrate and dilute urine? No, because they live where?
In the water.
In the water. So they got water all the time, they don't have to worry about it, right? Maybe if they're in salt water versus fresh water, brackish water, makes a difference. But the bottom line is fish tend to have these very short loops of Henle because they don't have to worry about water because they're in it. B and C, which one's going to be the kangaroo rat? C. C. Why? Because they live in the desert and they don't know when they're getting their next drink. Kangaroo rats can concentrate their urine to such a degree that literally they can pee crystals. They're designed to do that. Why? So they don't get rid of water in that environment. Humans are right in between. Matter of fact, we have some nephrons that are really long and most that aren't. About 10 or 15% of our nephrons tend to be really long, but the other, 80, 90% of them are relatively short. And so the ones that are really long are best for concentrating or diluting urine. And we're not going to talk about the way it does it, but in a brief sentence, the osmolarity gets more and more concentrated as you move down the nephron, and so that helps to pull water out. So when we talk about reabsorption, right, by the time we get to the end of the descending limb, most of the water has been reabsorbed that we're going to do. Okay, not all of it, but most of it. The loop itself just sends us back the other way. We don't have to worry about this. This is just called the loop. So not surprisingly, this side would be called the ascending limb, right? And the ascending limb is exactly opposite. The ascending limb is permeable to everything but water. Okay, so the ascending limb is permeable to everything but water. And again, this allows us to concentrate or dilute solutes without having to worry about the water flow. Everywhere else in the body, if water goes, solutes go, or vice versa. Here, we can reabsorb sodium as an example and not move water. So in the ascending limb, we're permeable to everything but water. And one of the mechanisms we'll talk about in terms of sodium balance is in the ascending limb, we have little protein reabsorbers that reach in and pull out sodium. And in this case, water does not follow sodium directly because it can't move across the wall. And again, it allows us to independently control solute or water concentration without worrying about the other one in each part of these limbs. Okay? The distal convoluted tubule is pretty important. And the distal convoluted tubule is primarily there for secretion. Okay, secretion. Secretion happens late in the process, which makes sense because when you're getting rid of stuff, you're getting rid of stuff you don't want, right? Like today's trash day. And my son, who was the age of many of you, actually took out the trash this week, so I didn't have to, right? But you take out the trash like Tuesday night when they come and pick it up Wednesday morning. Why? Because you don't have to deal with it, you know, in front of your house for a week. We do the same thing here. If we dumped, let's say, acid into the proximal convoluted tubule, what's going to happen to pH? It's going to drop, right, a lot. I have to worry about that pH drop and how do I protect the nephron as it moves through, right? If I dump it in at the very end, hey, I don't have to worry about it much because I dumped it at the very end. So secretion happens in the distal convoluted tubule primarily late. We secrete a number of different things and really depends on our acid-base balance. Sometimes we secrete hydrogen ion. Oftentimes we do because we make a lot of acids, we kind of talked about before. But sometimes we secrete ammonia. Sometimes we secrete nitrogen, sometimes we save it. There's not a whole lot of things we secrete, but when we want to, we can actively secrete things in the urine. We actually think of the digestive system as getting rid of waste. It doesn't, technically. The stuff that comes out the other end of your digestive system is stuff that you put in your mouth and never absorb. But we can't in our digestive system say, hey, we don't want this, and put it into your guts. What's in there is in there. If we don't absorb it, that's what comes out. The kidney's different. The kidney, we can say, hey, we don't want this substance. We're going to stick it into the urine, into the ultrafilter at this point in time, to get rid of it so we can pee it out. So we really do have the ability to get rid of waste, like xenobiotics, like penicillin and things, in terms of the kidney. Okay? So that's the secretion process. It happens primarily in the distal convoluted tubule. So that's kind of the purpose and the different parts of the loop of Henle and what each portion of the renal tubules do in terms of that. So make sure I talk about everything. Couple last things here. In the distal convoluted tubule, we have a couple important cells. Okay? So these are just, we're going to put them under the heading of cells of the DCT. The first type of cell they're going to worry about are called principal cells. So principal cells, like principal of a school. All right? So principal cells are very important in terms of how things work. And we haven't talked about it yet, but we have a hormone called antidiuretic hormone, or ADH. And they're sensitive to ADH. Okay? And principal cells have in them, which is, since we discovered them recently, the name actually makes sense. Okay? Inside each principal cell is what we call an aquaporin. Aqua sounds like what? Water. Porin, hole. They're literally water holes. They're holes for water. And what happens, and I don't know why students have this problem, but they have problems with what antidiuretic hormone does. You have no problem. You all know what diuretic does, right? So if you take a diuretic, what's going to happen? You're going to pee more. So if you take an antidiuretic, you're going to do what? Pee less, right? How do you pee less? You're going to reabsorb more water. So antidiuretic hormone allows you to reabsorb more water. And what happens is it opens the aquaporins in the principal cells, and so water that would be leaving through the distal convoluted tubule go through the holes and get reabsorbed. The peritubular capillaries pick that fluid up. Does that make sense? So, you know, that question we had earlier about how much water you reabsorb, people with that type of diabetes I mentioned that's not mellitus, they don't respond or make ADH, and they can pee literally 20, 25 liters a day in the worst case. Most don't, but that's bad cases. So principal cells sensitive to ADH, antidiuretic hormone, we'll talk more, and it opens aquaporins to help reabsorb water. There's another type of cell called intercalated. Some people say intercalated. The intercalated cells, their job, hydrogen ion. So they're the ones that reabsorb or secrete hydrogen ion, depending on what your acid-base balance is. So if you need to get rid of hydrogen ion, the intercalated cells just spit it into the ultrafiltrate to get rid of. If for some reason you needed to retain hydrogen ion, for your blood pH was too high, let's say you had alkalosis, right? In this case, you don't want to pee out hydrogen ion because that's going to drop your pH, right? So you're going to reabsorb it. And what they do is they reach in and take out the hydrogen ion before it goes out as urine and helps to balance pH. So the intercalated cells can do both. Get rid of hydrogen ion, take in hydrogen ion. Okay. It's a little more complicated than that, but that's probably a nice little summary of it. All right, so taking a quick look, we should see quick review. Got the renal artery. Eventually, you go through a whole bunch of other arteries, get to the afferent arterial. You have the glomerular capillaries. That's our portal capillary 1. Again, the job of the glomerulus is to filter, right? What doesn't get filtered comes out the efferent arterial. How much gets filtered? Of 100% in the afferent, about 80 or 85% comes out the efferent, right? Which means, where's the other stuff? It became ultrafiltered. Does that make sense? It became ultrafiltered. So, as an example, not a bad estimate of kidney blood flow. About 600 mils per minute. Not a bad estimate of how much blood flow goes through the kidney. So it's about 10-12% of your cardiac